An AI-generated post for your viewing pleasure. The future of neuroimaging. Unlocking brain activity with EEG to fMRI synthesis. Imagine capturing high-resolution brain activity in real time using nothing more than a low-cost EEG headset. New groundbreaking research proposes a revolutionary framework that bridges the gap between EEG and fMRI, paving the way for a future where monitoring brain health is more accessible and effective than ever before. Before we dive in, let's take a moment to understand what this research enables. We're going to explore how EEG and fMRI technology work and their respective roles in understanding brain activity. Welcome back to our channel. Today, we're diving into an exciting breakthrough in neuroimaging research that could change the landscape of how we understand brain activity and diagnose neurological diseases. The groundbreaking study we're exploring introduces a novel framework for converting electroencephalography, EEG, signals into functional magnetic resonance imaging, fMRI, detected blood oxygen level dependent, bold signals. Why does this matter? Because it addresses the high costs and accessibility challenges associated with fMRI, providing a means for improved clinical diagnostics and real-time monitoring of brain function. So, let's unpack what this means for the future of neuroscience. In the realm of neuroscience, understanding brain activity is crucial for diagnosing and treating various neural disorders. EEG measures the electrical activity of the brain with high temporal resolution, providing insights into how the brain reacts in real time. On the other hand, fMRI detects brain function through changes in blood flow, allowing researchers to visualize which areas of the brain are active during certain tasks, but suffers from lower temporal resolution. These two techniques each have their strengths and weaknesses, which often limit their standalone effectiveness in clinical and research settings. The research we are discussing today focuses on synthesizing bold signals from EEG signals using a framework called CATD, or condition-aligned temporal diffusion. This framework leverages the complementary strengths of EEG and fMRI, enhancing our ability to capture brain dynamics that were previously challenging to access. By bridging these technologies, the research highlights its potential for revolutionizing how we interpret neural activities and enhance our understanding of brain pathologies. Now that we understand what the research is about and its significance, let's delve into the technical contributions of this groundbreaking framework. At the heart of this research lies the CATD framework, which stands for Condition-Aligned Temporal Diffusion. This innovative method identifies a way to synthesize high-resolution bold signals using low-cost, readily available EEG signals. The framework utilizes two key components, the conditionally aligned block, KB, and the dynamic time-frequency segmentation, DTFs. 1. The CAB module functions by aligning the heterogeneous neuroimaging data of EEG and bold signals into a unified representation space, enabling effective cross-modal synthesis. This aligns with our earlier realization that the integration of disparate data types is pivotal in modern neuroimaging strategies, emphasizing the need for interoperability across various imaging modalities. By efficiently integrating these different signal types, CAT dramatically enhances the clarity and accuracy of results. 2. The DTFs module complements this process by harnessing EEG's superior temporal resolution to improve the temporal aspect of the bold signals, allowing the framework to capture dynamic brain activity changes that traditional methods might miss. The interplay between neuroimaging temporal dynamics and their effects on signal fidelity hints at exciting future research opportunities, especially concerning enhanced real-time monitoring capabilities for clinical applications. What makes this approach so revolutionary? It not only increases the accessibility of important neuroimaging data, but also enriches the quality of insights gleaned from these datasets. The implications for both research and clinical practice are profound. With those technical details in mind, let's now discuss how these contributions fit into the broader landscape of neuroscience research and the potential impact they may have on the field. As we dive deeper into the results from the study, it's impressive to see how CAT8 outperforms existing methodologies with experimental validation, indicating significant improvements in accuracy for identifying abnormal brain regions. This is especially promising for diseases such as Parkinson's, where timely diagnoses can make a world of difference. What's particularly exciting is that this research builds on the growing trend of using generative AI models for cross-modal data synthesis, tapping into their potential to unify disparate neuroimaging data into cohesive, actionable insights. While previous studies have hinted at the interconnectivity of EEG and fMRI signals, CAD-AI showcases a comprehensive solution, solidifying this connection and positioning itself as a tool for future neurological research. However, it's essential to recognize some critical challenges that could hinder the practical adoption of the KTD framework in clinical settings. For instance, the implementation may require specialized equipment and trained personnel, which could pose significant barriers to its integration into existing workflows. While the authors highlight the advantages of KTD, they do not sufficiently address these practical challenges, which could limit its utility in everyday medical practice. Moreover, the variability in EEG and fMRI data quality across different patients is another concern. The study's datasets are notably limited in size and diversity, comprising only a small number of subjects from specific demographics. This raises questions about whether the findings can be generalized to broader populations or different clinical scenarios. Without addressing the quality of input data, the reliability of the synthesized outputs remains uncertain, particularly in diverse patient populations with varying neurological conditions. In conclusion, while the KTD framework opens up exciting avenues for advancing neuroimaging research and clinical diagnostics, it is imperative that future studies address the practical challenges and ethical implications of using generative AI models. By doing so, researchers can ensure that advancements in the field are not only scientifically sound, but also ethically responsible, paving the way for a more trustworthy and effective application of neuroimaging technologies in clinical practice. As we continue to explore the intricacies of our brains, frameworks like KTD will play a pivotal role in enhancing our diagnostic capabilities, personalizing patient care, and fostering deeper understandings of neurological disorders. We look forward to seeing how this innovative research will evolve and influence future studies, especially as we strive for greater inclusivity in medicine by leveraging more accessible technologies for patient monitoring. Have any thoughts on how this could impact medical practices or research? Drop your thoughts in the comments below, and don't forget to like, share, and subscribe to stay updated on the latest neuroscience advancements.